Hi friends, I am Suhas KN, studying in second BSc Horticulture at Kolar. I welcome you all to this slideshow about biofertilizers. I am here to explain about biofertilizers and its types and their application methods and advantages and disadvantages. Let's start with the introduction. A biofertilizer is a substance which contains living microorganisms when applied to seed, plant surfaces or soil, colonizes the rhizosphere or the interior of the plant and promotes growth by increasing the supply or availability of primary nutrients to the host plant. Biofertilizers add nutrients through the natural process of nitrogen fixation, solubilizing phosphorus, and stimulating plant growth through synthesis of growth promoting substances. So we have bacteria, algal biofertilizers, phosphate solubilizing bacteria, fungal biofertilizers, earthworms. In bacteria, symbiotic nitrogen fixers like Rhizobium, Azospirillum species, and next free living nitrogen fixers like Acetobacter, Kelpicilla, etc. In algal biofertilizers, blue-green algae in association with azolla, first one. Second one, Anabina nasta acylatoria. In phosphate solubilizing bacteria, Pseudomonas bacillus megatherium will come. In fungal biofertilizers, VAM will come. Vesicular herpescular mycorrhiza. Next is earthworms. Earthworms also play a very important role as biofertilizers. Next, we shall deal with types of biofertilizers. In that, bacterial, fungal, algal, aquatic worms, and earthworms. Next, we want to know about what is biofertilizer. Biofertilizers are natural fertilizers that are microbial inoculants of bacteria, algae, and fungi, separately or in combination, which may help biological nitrogen fixation for the benefit of plants. They help build up the soil microflora and thereby soil health. Biofertilizers also include organic fertilizers like manures, etc. Use of biofertilizers is recommended for improving the soil fertility in organic farming. Next, we shall deal with bacterial biofertilizers. The live cells of bacteria used as a biofertilizers. These microbes contain unique gene called as NIF gene, which make them capable of fixing nitrogen. The nitrogen fixing bacteria work under two conditions. First one is symbiotically, next free living bacteria. The symbiotic bacteria makes an association with the crop plants through forming nodules in their roots. The free living bacteria do not form any association but live freely and fix atmospheric nitrogen. Free living bacteria. Large number of free living or non-symbiotic bacteria, that is, does not form nodules but makes association by living in the rhizosphere, that is present in soil. These are commonly used as free living bacteria, and these are classified as Acetobacter, Kelpicilla. Kelpicilla will not associated with plant. Astrobacter is a biofertilizer which provides the required amount of nitrogen to the plant from the soil. Next, we shall go through Azospirillum. It mainly present in cereal plants. It inhibits both root cells as well as surrounding of roots, and it forms symbiotic relation and increasing the nitrogen fixing potential of the cereal plant. Azospirillum is recognized as a dominant soil microbe. Nitrogen in the range of 20 to 40 kg per hectare in the rhizosphere in non-leguminous plant such as cereals, millets, oilseeds, cotton, etc. Considerable quantity of nitrogen fertilizer up to 20 to 30% can be saved by the use of Azospirillum in a plant. These species have been commercially exploited for the use as nitrogen supplying biofertilizers. Next is vesicular arbuscular mycorrhiza, that is VAM. The term mycorrhiza was taken from Greek language meaning fungus root. This term was coined by Frank in 1885. The mycorrhiza is a mutualistic association between fungal, bacilli and plant roots. VAM is an endotrophic mycorrhiza formed by acceptated Phycomycetes fungi. VAM help in nutrient transfer mainly of phosphorus, zinc and sulfur. Next is vesicular arbuscular mycorrhiza, that is VAM. The term mycorrhiza was taken from Greek language meaning fungus root. This term was coined by Frank in 1885. The mycorrhiza is a mutualistic association between fungal, bacilli and plant roots. VAM is an endotrophic mycorrhiza formed by acceptated Phycomycetes fungi. VAM help in nutrient transfer mainly of phosphorus, zinc and sulfur. Uses of VAM. It enhances the feeding area of the plant root as the hyphae spreads around the roots. It mobilizes the nutrients from distance to root. It stores the nutrients. It removes the toxic chemicals which otherwise hinder nutrient availability. It provides protection against other fungi and nematodes. It increases growth rate in plants such as citrus, maize, wheat, etc. It reduces sensitivity of crop towards high level of salts and heavy mechanism of action. The VAM forms an association with the plant root. It penetrates in the root cortex and spreads around the roots of the plant. As the name indicates, they possess sac-like structures called vesicles, which stores phosphorus as the phospholipase. The other structure called arbuscule helps bringing the distinct nutrients to the vesicles and root. Mycorrhiza is the symbiotic association between plant root and soil fungus of the certain types of mycorrhiza. VAM plays a great role in inducing plant growth. VAM are symbiotic endophytic soil fungi which colonize the roots of approximately 80% plants. The VAM hyphae also help in retaining moisture around the root zone of plants. It increases the resistance of resistant to root-borne or soil-borne pathogens and nematodes. Let's see about carrier material. The use of ideal carrier material is necessary for the production of good quality of biofertilizers. Peat soil, lignite, vermiculite, charcoal, rust mud, homemade manure and soil mixture are used as a carrier materials. Neutralized peat soil or lignite are found to be better carrier materials. Ideal carrier materials should be cheaper in cost, locally available, high organic matter content, no toxic chemicals, water holding capacity of more than 50%, easy to process. Next is Astrobacter. Astrobacter is a heterotrophic free-living nitrogen-fixing bacteria present in alkaline and neutral soils. Astrobacter is the most commonly occurring species in arable soils of India. Apart from its ability to fix atmospheric nitrogen in soils, it can also synthesize growth-promoting substances such as auxins and gibberellins and also to some extent the vitamins. It is the azolla as a biofertilizer. Azolla is a tiny freshwater fern common in ponds, ditches and rice fields. It has been used as a biofertilizer for rice in all major rice growing countries including India, Thailand, Korea, Philippines, Brazil and West Africa. The nitrogen fixing work is accomplished by the symbiotic relationship between the fern and blue-green algae, Arabina azolla. In addition to nitrogen, the decomposed azolla also provides potassium, phosphorus, zinc and iron to the crop as a biofertilizer. Another group of free-living nitrogen fixers are cyanobacteria, commonly called as blue-green algae. More than 100 species of blue-green algae can fix nitrogen. Nitrogen fixation takes place in specialized cells called heterocysts. PGA, very common in rice fields, blue-green algae. Unlike Astrobacter, blue-green algae are not inhibited by the presence of chemical fertilizers. No chemical fertilizers added. Inoculation of the algae can result in 10 to 14% increase in crop yield and will provide maximum number of population of each bacteria required for better results. Mixing of any of two bacteria and the treatment of seed will not provide maximum number of bacteria of individuals. Next is biofertilizers application methods. There are three ways of using these nitrogen fixing bacteria. These are seed treatment, root dipping, soil applications. One is seed treatment. Seed treatment is a common method adopted for all types of inoculant. The seed treatment is effective and economic. Seed treatment with Rhizobium, Astrobacter, Azospirillum along with PSM. Seed treatment can be done with any of two or more bacteria. This has no side effect. Important thing has to be as the seed must be coated first with Rhizobium or Astrobacter or Azospirillum. When each seeds get a layer of above bacteria, then the PSM inoculant has to be treated on outer layer of the seed. Next is root dipping. Application of Azospirillum With the paddy vegetable plants, this method is needed. The required quantity of phosphorus film has to be mixed with 5 to 10 liters of water at one corner of the field, and all the plants have to kept for minimum 1.5 hours before sowing. Third method is soil application. PSM has to be used as a soil application. Use 2 kg of PSM per acre. Mix PSM with the 400 to 600 kg of cow dung along with half bag of rock phosphate if available. The mixture of PSM, cow dung and rock phosphate have to be kept under any tree shade or ceiling for overnight and maintain 50% moisture. Use the mixture as a soil application in rows or during leveling of soil. Next, we see the advantages of biofertilizers. Renewable source of nutrients. It sustains soil health. It supplements the chemical fertilizers. Replace 25 to 30% chemical fertilizers. It increases the grain yield by 10 to 40%. It decomposes plant residues and stabilizes CN ratio of soil. It improves texture, structure and water holding capacity of soil. No adverse effect on plant growth and soil fertility. Next is disadvantages. Biofertilizers require special care for long-term storage because they are alive. It must be used before their expiry date. If other microorganisms contaminate the carrier material or if grower use the wrong strain, they are not as effective. Biofertilizers lose their effectiveness if the soil is too hot or dry. Thank you for watching video.